Okay, so good morning, everybody. Welcome to the GBATnet My Field for Dummies webinar on immunology. Today, we will have one of our student reps, Thongthai Thavornwatanayong, or Thai for short, who will be presenting about it. And his background is in immunology. So hopefully you all learn something. And, please remember to mute your mics. Questions can be at the end after he finishes. And Thai, I will hand it over to you now.
Thank you, Susan. So let me maximize this in presentation mode. And with that, I will get started. So just to give you guys an agenda, what we'll be talking about today, I'll just introduce my journey into immunology first, and then we're going to start talking about the basic components of the immune systems, especially the mammals, because that's what we're interested in. We're going to then talk about some of the techniques that we utilize in immunology that you're going to find in a lot of the immunology paper. And from there, we're going to make a transition from the model organism, which is mice, humans, into more of the bat immunology, which is why a lot of people are here for. So with that in mind, I will tell you about my journey into immunology. It started off in bachelor, where I was doing my degree in biochemistry. I wasn't sure what I wanted to do, and then for one of my class, I read a book called Spillover. If you have read it, you know what it's about. If you don't know, it's about zoonosis, and it talks a lot about the immune systems and the component of the immune system that are involved in the epidemics or pandemics. So that's how I first started to be interested in the immune systems. And from then on, that led me to pursue a degree in biotechnology at the Biomedical Science School at UCF in Florida specifically. That's where I was able to work in lab. So I was in the T cells lab, which is that's one part of the immune system. And then from then on, I make a transition into a tech at the innate immune signaling. That's specifically for the asthma related pathways. And then I got kind of bored of mammals, so I took some time off to go back to Thailand, and I still want to stuck with the immune system. So I study invertebrate immune systems, specifically in the shrimp. And after I finished my master's, I went on to be a technician at the cancer center at Einstein. That's where I do some research on the colorectal cancer treatment, and it ended up still utilizing a lot of immunology concepts. We talk a lot about the immunotherapeutics in cancers and so on. So I'm still kind of in the realm of the immune system, but more on the application side. But I kind of got bored of that. I really want to understand just the basic fundamental of how the immune system works. So when I start my PhD, I apply for the lab that do that. And because I haven't really worked with B cells before, I ended up joining Dr. Wong's lab at City College, who where we study the B cell and immunoglobulin and how it's being regulated. And it's also the same place where I met Susan, who now is my co-advisor, and we're studying bat immunoglobulin. That's how I get into the bat world. So what exactly is immunology? Basically, it's just the study of how the a particular components of the immune system individually work or interact with each other in response to antigens. What are antigens? The antigens are the molecules or substances that can elicit immune response. So that term will get thrown around a lot. So I will clarify what they are now. A lot of the information that I get from here are based from this textbook here, from Weds Immunology. That's a little advanced, but it gives you a very good details of every specific part of the immune system. And if you a little bit intimidated, you want to start somewhere a bit easier, or just want to read about immune system like recreationally, I recommend this book called Immune. It has a lot of nice picture and less words, and maybe you can start from there. And then if you find particular aspect that relates to your research or you're interested in, then you can go into the Ways and look at that specific components for more details. So the immune system for this one particularly is the mammal immune system. consists of the two arms. So you have the innate immunity and adaptive immunity. The innate immunity are very fast response. They are the first line of defense against the invading microbes. So the foreign microbe that can cause the disease, we call those pathogen. So basically the innate immune response recognize the broad pathogens that is different in adaptive immunity, where it's more delayed response. It's not the first line of defense, but it take longer to mount the immune response, but you ended up with something that more specific against that invading microbes, and it's also longer lasting. So it's more time to invest into building up that immune response, but you have something that can be very effective against the pathogen. Let's see. Oh, okay. So with that in mind, we jump into the innate immunity. The true first line of defense in the innate immunity is the natural barriers. What are these? Well, these are the physical barriers, such as skin or mucosal layers in the intestine. What picture is showing here is the lining of the intestine that preventing the microbe from getting in. On top of being just the physical barrier, you also have the chemical barrier, which is this yellow layer that's showing here that kind of coated the epithelial layer of the intestine. These chemical barriers contain specific components that are either like toxic against the bacteria or make it less likely the bacteria will hang around. So you have something like lysozymes. These can also be found in tears. That can digest away the proteins that and kill the bacteria that way. You also have antimicrobial peptides, and beside these components, you also have things like stomach acid in particular tissue. That's already like a less desirable environment. It's a harsh environment for certain microbes to survive. So with these are the first like protective barrier is the wall. It's preventing your inverter from coming into the system. However, if they do getting in, we do have things that work for that. So the first thing that will work is the pattern recognition receptor, or PRR. And these receptors come in many flavor. Don't worry too much about the names. The idea is that these receptors recognize specific particular type of molecules, such as liposaccharides or the protein that are found in the microbes or the pathogens. And the term that we use to describe these molecules are called pattern-associated molecular patterns, or PAMs. Beside PAMs, you also have a different version of this called DAMs, or the damage-associated molecular patterns. Instead of you having molecules coming from the pathogen itself, DAMs are just a piece of dead cells, usually from the infected cells that dying. Once the cell die, we release some of these molecules that can be recognized by these receptors, and that lead to the downstream illicit like induction of the immune response. The way that I like to look at the pattern recognition receptor, I see them as a sensors and alarm. When they sense some sort of the sign of the microbes or antigens associated with the microbes, they will ring the alarm to bring in other downstream immune response to work against it. So beside this, you have the proteins that kind of linger around. The complement system is what we call it. The complement system is pretty primordial. You can find them down to like invertebrate. Essentially, they are the protein subunit that recognize specific pathogen through different means. And there are three different ways to recognize that. You have the classical that need antibody to mediate the recognitions, and you have the some that can just bind to the microbe directly, such as lectin, that can just bind to the bacteria based on the liposaccharide molecules or something like that. And alternatively, you have the slightly different version of that recognize fungus. It recognizes specific molecules that present in the fungus. But what all of these have in common is that in recognition of those microbes that it bind to, it's gonna lead to the downstream signaling cascade that going to induce specific immune response like phagocytosis. We will go into detail what that is, or recruitment of the immune cells. That's what inflammation does. And or you can just build some sort of a subunit on the microbes and kill them by lysing them. So I talked about the sensor and alarm. What are the soldiers and the player that come in the innate immune system? And this would be your innate immune cells. I have some of the example here. You have something like macrophage or neutrophils that can do multiple things, and I'll go into details of like what this does. And you have something like NK cells and dendritic cells, and these are just the name. Don't worry about that too much yet. But the big thing is that the innate immune cells can be divided into three main functions that they do, which include phagocytosis, or releasing of like the chemical to kill the microbes, or presenting the antigen to help stimulate the adaptive immune response. And we'll go over each one of these functions, starting with phagocytosis, which is literally translated from Latin for cell eating. So some of the innate immune cells, like macrophage and neutrophils, can kind of survey around the tissue, like just epithelial tissue, what the microbe would get in. If they find the microbes, they can just eat it and digest it away, and then just spit out whatever left over in a soluble form, so it doesn't linger around your systems. That's the first function of the innate immune cells. The other one is the releasing of the chemicals, and I just call it chemical assault, just because that's easy to remember. But basically, certain innate immune cells contain granules, which is why you might hear the term granulocytes in some of the paper. It's just a way to describe that these cells have like little pockets inside the cells, little vesicles inside the cell that contain chemicals. And if some of these immune cells can eat the target, they will release the chemicals onto at the target. So depicting here in this picture is the neutrophil that releasing these purple dots, which are the like the granule components that is toxic to the pathogen. In this picture is the virus. So if you can't eat it, you just kill it. That's how I see it. And then some of these cells you might heard of, all the neutrophils, basophils, eosinophils, you might not have heard a lot, but mast cells is something that you might heard of a lot because it's associated with the allergy. When you have allergic reaction, it's the mast cells releasing histamine. So that is it doing that to the release of those the chemicals. So beside targeting the microbes itself, if it's able to detect microbes, it can process these microbes into specific antigen to stimulate adaptive immune cells. In this case, it's the T cells. So the adaptive immune cells require the antigen presentation in order to be stimulated. The T cell is particularly picky. They only get activated when the antigen is presented on the MSC molecules. And the MSC molecules, I see them as a silver platter that like the T cells won't take in any antigen unless it's being presented this way. So they a picky eater. If you don't serve it on the nice platter, it won't get stimulated. So the picture here is the dendritic cells. It is a well-known innate immune cells with antigen presentation functions. And presenting the antigen to adaptive immune cell is also what I see as a link between the innate immunity and adaptive immunity. So with that in mind, we're going now jump from the innate immunity into the adaptive immunity. The adaptive immunity, I'll remind you what it is, like what the characteristic is again. So the main characteristic of the adaptive immunity is that it's specific to the antigen, and that is possible because these immune cells and molecules contain the antigen receptor, such as the T cell receptor in the T cell, or B cell receptors in B cells. So they can recognize very specific pathogens, and do antigen presentations, they can make a bunch of these immune cells that target that specific antigen. So it's more a little bit more specific than the innate immune cells. It's more honing in on specific target. So it's a little bit more effective in that sense. So the cells in the adaptive immunity, T cell and B cells, some people might have heard of it. If you go to the hospital, sometimes they look at that to check up on your health. And there are also proteins, which is released by B cell called immunoglobulins, also known as antibodies. You might sometimes hear them being used interchangeably, but in some of the immunology paper, they use the term immunoglobulins rather than antibodies, to not mix up the reagent used in the study versus the actual molecules that we study. So T cells can be differentiated into two main groups. You have the helper T cell, regulatory T cells. These are known, they have specific marker, CD4. They don't directly interact with the microbes, but they stimulate other immune cells. So one example that's showing here is the helper T cells, stimulating B cells to activate. And this is a typo, it's supposed to be B cell activation. My bad on that. But T cells can activate B cell to produce the immunoglobulins that can help with the fight against the invading pathogens. The key takeaway, again, within adaptive immunity, everything kind of require antigens. So the T cells stimulating B cells require B cell to present this antigen, this red dot here, to T cells, and in addition to some of the signaling through receptor ligand interaction in order to get activated. So that's one of the group of the T cells, and what it does is stimulate other immune cells. Besides this, some T cells can actually use their antigen receptor to recognize the infected cells and kill them directly. And we call this cytotoxic T cells because they kill the cells. These are known. Sometimes you will hear CD8 T cells because these T cells express CD8 on the surface. That's just the surface molecules. That's how you kind of differentiate it from the helper from the killer. So now, I mentioned the T cell can activate B cells. What does B cells actually do in the adaptive immunity? Well, the main function that people know is that it produces immunoglobulins. When B cells get activated, it's going to become differentiated to the plasma cells. And when it becomes plasma cells, it can release immunoglobulins that can target microbes. And I'll go over how they do that. So before we go into how immunoglobulins fight against the pathogen, we have to know just the main portion of the immunoglobulins. So the immunoglobulin contain two main portions. You have the variable regions that are going to recognize and bind to the antigen, and you have the constant region, which is down here, that can determine the functions utilized to fight against that pathogen. So these functions, I kind of group them into three main groups, but I'll go over one by one. So the first one straight up is that it can just bind to the pathogens, and by binding to that pathogen, it preventing these possibly pathogenic microbes from spreading around and infect the nearby cells and so on. Alternatively, it can facilitate phagocytosis, which I mentioned before. This is something that innate immune cell does. It does it by opsonizing the target. This is also the term that you see in the immunology paper. Opsonization, it's just a more complicated way to describe coating the target with the immunoglobulins. by coating the target with immunoglobulins, it facilitates the innate immune cells to easy like eating the target easier or recognizing the target they need to be eaten. Beside this, coating the target can also help bringing the killer cells to the target to kill that cells. And in addition to all of this, it can also mediate the complement pathway. So binding to target can recruit the first component required to activate complement pathway, which I mentioned before, bring about other immune response. So immunoglobulins come in many flavors, and we call these different isotypes. These different types of antibodies have different constant regions, which I'll remind you is just this bottom portion. And because the constant region determines the function of the immunoglobulins, some like each type of these immunoglobulin gonna function slightly differently from each other. And that's it's nice in the sense that you can kind of fine-tune the immune response that would work best against particular pathogens based on what type of immunoglobulin the B cells can produce. In addition to the innate adaptive immune component that I mentioned before, you might have heard that of cytokines. I put this outside of innate and adaptive because both innate immune cells and adaptive immune cells, also some of the non-immune cells utilize this for cell-to-cell communication. In the context of immunology, cytokines help recruiting the immune cells to the target, stimulating them to differentiate into specific type of the immune cells depends on what type of cytokine stimulated them. And that allow for making kind of like mounting the specific immune response against the target. And it also can also be involved with the chemotactic. That's the subtypes of the cytokines called chemokines. These are mainly just help with the cell migration to the target. So that's involved with the recruitment of the cells to the target. The example of the cytokines I will bring up is the interferons. And I bring that up because we talk a lot about interferons in the bat and how it involved with the antiviral immunity. So I mentioned each of the components, but they don't necessarily work by itself to clear up the invading microbes. Basically, immunologists study how each of these components work in response to the target. In this case, it's the coronavirus. This is not necessarily drawn to the scale, but the picture showing here, it's just to depict that you have both the innate immune component that responds to the pathogen, and you also have some of the adaptive immune response, adaptive immune components that respond to the target, and they all work in coordination with each other, which is why it's a system. So showing here just macrophage that require antibodies to help mediate either phagocytosis or releasing of the chemicals to kill the target. And you also have some of the B cell and T cell that in presence of the antigen, they'll just stimulate the either cytotoxic T cells immune response or make the antibodies to work against the target. So understanding how the immune system work, how all of these components work together, allow us to utilize that knowledge for things like therapeutics. So the example that I bring up is COVID-19, just because we just got, we're still somewhat in the pandemics, but most of us have the vaccines. But in order for the vaccine to develop, we need to understand how the adaptive immune system work to really make the best vaccine to mount immune response against the virus. Alternatively, you can also mount the immune response against the non-pathogens. In this case, like cancer cells. That's the immunotherapy. You can boost a patient immune system to recognize the cancer as the as the non-self foreign components that immune system can target. So you can kill the cancer with your own immune system in this way. So besides therapeutics, you can also utilizing the knowledge immunology for the diagnosis. For example, if you understand for the particular with disease patient showing in red here, they might have some of the mutation in certain immune components or the defect in certain immune components when compared to the healthy individual, which is shown here. And these mutation could be this red bar that's showing here. Identifying this target allow you to know how what could be underlying these diseases, and also identifying these patients for particular disease, and understanding that allow you to utilize the type of therapeutics that can help treat the patients. This is more on the individual scales. At the larger scales, understanding immunology and immune systems and the components within the immune system can allow you to assess the herd immunity. So example of this is this paper that talk about how different persons have a slightly response to the vaccine. Like the vaccine is not effective on everyone, but like each individual is slightly different from each other because of their they have a slight difference in the immune components. Understanding this at the population scales allow us to understand the population susceptible for certain diseases like some zoonoses, or it also allow us to assess how certain therapeutics might be effective within the population to fight against the disease. So now that I talk about applications, we're gonna come back to how we actually understand immunology. So how do people actually study immunology in labs? What techniques do they use? Well, I kind of list out the key things that you can find in a lot of the immunology paper, and that's involved just the induction of immune response, and specific assay to assess the immune response, and that is enzyme-linked immune sorbent assays, ELISA, or the flow cytometry. So to really understand immune system in the model organism like mice, you first have to induce that immune response, and you can do that by two different way. You either use specific antigen, so it's just the specific molecules that induce the immune response, that not necessarily the actual pathogens or the microbe itself. An example of this are liposaccharides molecules from the bacteria, or the protein from different species, such as chicken gamma globulin. When you put that in mice, they will elicit the immune response, because that is the mice recognize, the my immune system will recognize that as the foreign molecules. Beside that, you can also just directly throw the pathogen into the mice. Example of that I listed here is influenza A. And I like that that particular microbe because it is used a lot to study influenza, immune response against influenza, particularly because the influenza A only infect mice and not human. So you can work with mice and not having to be afraid that you're gonna get sick somehow. But this is not always doable in the field. Of course, in the field, you don't have the control environment. You work with wild animals that been exposed to all kinds of things, um, all kinds of microbes. So the only way that you could really study that is just to do some comparative analysis between the animal that have the sign of disease that would be indicative of having the immune response against that disease, versus comparing that to the healthy individual animal. And that's really like the only way that you could do in the field, if you can't challenge the animal in the field, or adding in the antigen into the animals. So after you induce the immune response, you can assess the immune proteins through the technique called ELISA. And ELISA involve the use of antibody, which is showing green here. So you can have the target protein coded onto the well, and with the antibody specific to that protein, you can capture that target. This antibody is enzyme linked. So when you add the substrate into the mixture of the antibody binding to the target, you can produce a signal. Sometimes it's the color change, sometimes it's the luminescence, depends on the substrate and the chemical reaction involved. And this is directly correlated with the amount of the proteins, because the amount of protein will determine how many antibody can capture that target and how much signal going to produce. So it's a good quantitative method to quantify proteins. In immunology and in the clinics, the protein that we usually look at are from blood, specifically the serum in the blood, that might contain proteins like immunoglobulins. So with ELISA, you can measure the titer of immunoglobulins in individual after the immune response. But one thing to keep in mind is that when you collect the blood at a specific time, you take the picture of the snapshot of the immune response. So if you want to see how that immune response changed over time, you would have to collect a time course samples and look at all of each time to understand the dynamics of how much of that immune proteins change in the system. So I mentioned ELISA to detect proteins. For immune cells, you can use a technique called flow cytometry, which is also antibody mediated. It's still dependent on the use of the antibody. And the flow cytometry, how it works, the simplest way that I can just sum up how it works, it utilizes it involving blood and cells. So I'll just walk you through how that works. So for example, you want to see how much of the certain immune cells population are in the tissue, you're gonna crush that tissue to release a bunch of the immune cells that you're going to resuspend in the blood. Let's say that the target immune cells that you want express this blue marker on the surface. You can detect these cells using the antibody specific to that marker. And for flow cytometry, we use the antibody conjugated to the fluorescence. So we're detecting these cells based on the fluorescent signals. So after you kind of probe these cells with the antibody, you're going to run this whole mixture of the cells and the antibody through the machine, which is going to flow the cells through a channel one by one. As it passes through this channel, if it had the fluorescent, it's gonna produce the specific fluorescent color. So this way you can kind of quantify how many of the cells of interest that you have. And beside looking, seeing that marker that correlate with the fluorescent signals, you can also determine the type of cell sometimes based on their complexity and size. And this is based from another light beside that laser that excite that fluorescent. This light going to scatter based on the cell complexity and the size. So besides just detecting the cells, there are different modification of flow cytometry. And then this modification allow you to actually separate out the cells that have the marker of interest, like the self-interest, into out of that whole population. And sometimes people can culture that cells or study that cell specifically. So I talk a lot about these immunology techniques and what we know about immune system, but these are all from the mice. We're here for bats. What about bats? And this is where we're gonna make a transition into the bat immunology. To talk about bat immunology, I'm gonna talk about three main things. why do we even want to know about bat immune system? And then what do we actually know about bat immunology so far? And then what are some of the challenges that involve in studying bats? So why are we bothering study bat immune system? We know as people to study bat that they are highly diverse taxa, and then you can find them in all over the globes except for Antarctica. They also have unique physiology. They're the only mammal that fly. There's some sort of thing that involve the flight adaptation. And beside this, we know that some bats are associated with zoonosis. Example of that would be some flying fox associated with Henipa viruses, or Rosetta, or the vampire bat associated with the rabies. So the immune system, as I mentioned before, involving defense against pathogen. So in a way, it might fit with how bat adapt to particular environment that may have different pathogens. So understand immune system might help us understand why are there such diverse distribution of bat all over the globes. And it can also help us understand how bat adapt to flight, how bat adapt their physiology to flight, because there are link between physiology and immunity. And beside this, understand bat immune system, which just allow us to understand why are some bat associated with zoonosis, why are some bats can carry certain viruses, and not really being affected, really be impacted the same way as other mammal species that would be affected by the viruses otherwise. So with that in mind, we're going to talk about what we know about bat immunity so far. And this is going to be very brief because this is a 101 course. And I divided what we know mainly into the three main components I presented before, which is innate, adaptive, and auto-mediated, which is specifically cytokines. So we know that there's a positive selection for certain antiviral pattern recognition receptors, so that TLR8 or RIG-I, and these are the receptors specific to the antiviral response. In terms of the adaptive immunity, we don't know a lot about their adaptive immunity. Our knowledge is very limited. We know that they have T and B cells, like mice. We don't completely understand how they functions similarly or differently from the mice. They have immunoglobulins, but their immunoglobulin response, it's slightly enigmatic. For some of the study that have challenged the bat with either viruses or antigen, they found that the immunoglobulin response is lower in bats compared to like some other model organism like mice. And we just don't know why or how, and that's just the gap in knowledge that we have to explore. Beside this, the specific cytokines has been shown to be expressed in response to the antigen stimulation. And in this case that I bring up is poly-IC. Poly-IC are the synthetic nucleic acids that induce the response similarly to the idea is the poly-IC kind of similar to the viral RNA. So you can use this genetic molecule to kind of mimic the antiviral response. And when you stimulate the bats with these antigens specifically, they found that there's upregulation of type 1 interferons, cytokines. And just to remind you, this help in coordinate the immune system. The type 1 interferon specifically related to the antiviral immunity. So a lot of study has been going toward bat antiviral immunity, but we still don't know enough to understand how this process work in the bats compared to mice. So why do we don't know a lot about bat immunology? Well, because studying bat immunology is not easy, and there's challenges that I group into three main categories. The first one being technical feasibility, especially in the field, and there's the lack of the baseline information, the foundation knowledge required to do any downstream study, and there also just the lack of other resources required to do the type of the immunological study in the bats. Technical feasibility, first of all, some bat species are just endangered, some are protected, so little samples out of questions. And if we can collect the little samples, that limited us to look at only specific part of the immune system, like in blood, which is just the circulating components. This component will be circulating in the blood, but we won't understand how the B cell, T cell stimulations, and so on actually happen. That requires to look at the tissue, like spleen or lymph nodes. And those are the little samples. So some of the species we can't even study that. If we're able to collect those samples, there's another challenge in terms of the collection and preservation of the sample. The first one being that the type of the immune cells and the immune tissues are very fragile. You have to collect them in a time-sensitive manner, and to preserve them, you have to preserve them in the cold. The best way to preserve them is liquid nitrogens. And we know that liquid nitrogen is hard to access in the field. The picture showing here was from my Belize trip. These are the liquid nitrogen tank, like super bulky tanks that people have to bring in to be able to keep that sample cold. And then you have to figure out how to ship this back to whatever facility that you can work with. That brings me to another problem in terms of having the facility to study immunology, particularly in the field. You don't have a lab nearby that you can just do like sterile work, or having the machine like flow cytometer that you need to analyze the immune cells components. So these are the three main challenges in terms of technical feasibility. So other than being technically difficult, bats are not model organisms like mice. The model organism allows to understanding a lot of things about immune systems. Bats not being the model organisms mean that there are a lot of part of the immune genes and immune system that's not well characterized, and then you also have more variability in the wild animal versus something that you would have in the lab, like mice that you can breed for generations and still maintain genetic homogeneity. And besides this, we don't even know how healthy bat actually truly look like. We don't have a standardized way to assess if bat is actually healthy or sick or like mildly sick. And everyone kind of assessing at the moment, everyone kind of assessing that in their own way. But because we don't have that base information, we couldn't make that comparisons of sick animals that might have the immune response against that particular disease versus the normal immune response in the healthy individual. And beside all of this, there's also the question of the accuracy of taxonomy. In particular, if you want to do comparative works, you have to understand the evolutionary relation of the bats as a baseline to understand how how genetically different they truly are from each other, to understand how maybe immune genes might be different or the same in terms of the degree of different and similarities between species. And beside this, the current genome of the bat, we don't have the genome sequences of all the bats. And for the one that we have, it may not be annotated in the way that allow you to do the type of work that we can do with mice. An example of that would be immunoglobulin genes that I work with. I am required to know specific regulatory region in the genome, the non-coding regions that is not annotated or made publicly. And because I don't understand that, it's harder for me to understand the process that regulating immunoglobulin genes without any knowledge of the genomic backgrounds and so on. So it's a bit harder for me to studying that immunoglobulin because of that. And I'm bringing the local BAT1K because while there's this challenge here, um, there is some database that contain all of these genome sequences that eventually going to be made public. So that might help with the issue a little bit in the future. So beside liking the baseline informations and technical difficulties, we also don't have best reagents to study that, especially for the immunological studies, which I mentioned before. I brought up ELISA and flow cytometry. Both of these techniques require antibodies. And at the moment, I don't know if we have good monoclonal antibodies. Monoclonal antibodies are the antibody um, that target that's specific to just one target. So it's cleaner. It's a very clean antibodies. And I'm not sure if we have um, that monoclonal antibody that worked really well. That's one monoclonal antibody that I could find, and it was generated from the big brown bats. But I'll note that this was generated from total spleen cells. And if you use just total cells of the spleen, you're going to have all kinds of like antibody to target different immune cells. So I don't think we're at the level where we can sort out specific immune cells the same way that we could do in the mice yet. And besides not having these reagents, we have a harder time try to keep bats in captive to do any sorts of the manipulation with the animal to study it. Bats are super expensive to keep. They require special diets. They also require large enough facility because they do have to fly around. From what I know so far, there's not a lot of facility that contain bats. There's one in Israel with the Egyptian fruit bats, if I remember correctly. And that's one in Singapore for the Eonicterus, the nectar bat. And even with that, having that facility, the animals are not easy to produce. Like mice have a fast turnover of the offspring. Bats take a while to produce the offspring, and they only produce maybe like 1 to 2 for most of the species that I know. They don't produce a lot of offsprings, and they take a very long time to develop into an adult. So that's also more difficult. Why is it so difficult to work with bats? Um, with those challenges, some effort have been we are starting to try to tackle these challenges, and GBATnet kind of help with that because we are the network of the network that bring in people work in multiple disciplines and different regions together. We have the different working groups that assess different part of the bats. For the immunology, there's an immunology working group, and our objective to study bat immunology is to really understanding how the bat immune system kind of fit in with the bat diversity and diversification. And then you have the group that try to one way to sum it up, just understand what's actually healthy bat or what is stress bat. What these groups have in common is that we try to share information, eventually try to generate some sort of a standardized method to assess maybe specific markers for the immunology markers or the stress markers in the bat to have a better way to assess healthy versus the not healthy bats. And also sharing the methods and protocol to studying bats and processing the sample and collections. So with that in mind, I am now ready for questions. Thank you so much for your attention. I will take the questions now. So
Yeah, so if anybody has any questions, feel free to raise your hand or type it in the chat, and Ty will get to answering them. If there are no questions, though.
If you have questions later, my contact information are here. So that's my Gmail, and I'm also on Twitter. I check Twitter once a week. I don't check it as often, but it's there if that's easier for you to contact me.
Yeah, and Ty is involved also in the immunology working group. So if you have any questions about the working groups also, feel free to ask. Or any of the other efforts being made by GBATnet related to this. We're open to any questions.
Okay. Oh, sorry.
There are no questions.
No
Okay.
questions. Okay.
Well, Ty, could you tell us a little bit about the objectives of each of the working groups that you mentioned?
So for the immunology working groups, our main thing it's I think we want to utilizing different aspects to assess to understand bad immunology, such as like the genomics components, linked with some sort of like ecologies, um, ecological data, and maybe adaptations. We try to basically build pieces pieces what data that we have or data we could generate to understand bad immune systems, and what we have going on right now in terms of what we're trying to figure out what to do. I think we talk about focusing from just help tackling to the challenges required to challenges to the challenges that hinder our study about immunology. So for example, the sample collection and preservation, there is no standardized way that people collect the samples and preserve them, or it's not made publicly available. I know that Bat1K has it, but you have to be a member to access this. So with what we're doing is that we're thinking about generate that guideline or not guideline per se, but a protocols that everyone can like access to help with understanding like a better way to collect the samples and preserve them. And um I think we also talk about the sample sharing, which is we know that lethal samples are hard to come by. So we talk about sharing the samples, possibly or like a way to share the sample to see what everyone has. Um, do that. And the straight working group, I I haven't attended as many meeting in that group because I'm not part of that group. But as far as I understand, is that they and you can talk to the people from the stress working group more to understand what they're doing. But I know that they're talking about generating some sorts of the biomarkers that could be helpful to assessing stress in bats. And stress in bats can be a bit more broad. You can have the pathogens or like the stress that come from with the pathogen, or you can have the other environmental factors like abiotic factors that like impact bat stress. So those are kind of like general idea of these working groups.
Give me one second.
Okay.
Does anybody else have any other questions for Ty? If not, feel free to reach out to us at any time and just you have Ty's contact info. Yeah, thank you, Ty.
Oh,
I just want to make sure we end on time at 10, so I don't want to get too involved in any questions. So one quick question from anybody is okay, but no extended things. Please email Ty just to follow up. And also this is recorded, so it'll come up on the YouTube for GBATnet later, and you will also be able to find the slides on our website afterwards. Okay. Thanks for everybody for attending.
Well, thanks everyone for attention, and thanks for coming again. Please attend other My Field for Dummy in the futures if you're interested.
Yeah.
Okay, bye.
Bye, everybody.
Stop sharing.